Christianson syndrome is a rare X-linked neurodevelopmental disease, which is characterized by intellectual disability, epilepsy, ataxia, and mutism. But of most importance to our group, these patients also display a hyposensitivity to pain. With our multidisciplinary team composed of Dr. Reza Sharif Naini, Dr. John Orlowski, and Dr. Catherine Ferland-Legault, we elected to take an integrative approach to investigate the basis for this hyposensitivity. Notably, hyposensitivity to pain is one of the least investigated side effects of CS. However, it is one of the greatest concerns that parents of these children face when raising them. We provided the parent members of the Christianson Syndrome Association with surveys to fill out to describe the high pain tolerance that their children experience in greater detail. Parents described finding old injuries in their children that they had never even noticed, simply because their child never reacted or acknowledged this injury. CS is caused by various inherited and de novo mutations in the SLC9A6 gene that encodes the Nhe6 protein, of which over 60 CS-causing mutations have already been identified. Nhe6 is a sodium proton exchanger involved in controlling endosomal pH, which is ultimately important for regulating the recycling function of these cellular compartments. In order to have a mouse model of CS, we used an Nhe6 knockout mouse line which exhibits similar social deficits as those seen in human CS patients. To investigate the neural basis for the pain hyposensitivity seen in CS patients, we first characterized the neuroanatomical expression patterns of Nhe6 across different neural regions that are responsible for sensing pain in wild-type mice, and then we investigated behavioral changes in knockout mice in response to painful stimuli. Finally, we characterized the functional differences of their nociceptors using calcium imaging. This work was, of course, a group effort and was completed by Dr. Hugues Petitjean, Tarheen Fatima, Stephanie Mushbahani-Constance, and Albena Davidova from the Sharif Naini lab. Our neuroanatomical investigation first focused on characterizing the expression pattern of Nhe6 across different neural regions responsible for sensing pain. Surprisingly, we found that Nhe6 was significantly expressed in the dorsal root ganglia, but not in the dorsal horn, and only partially expressed in brain regions associated with pain. Upon further analysis of co-expression patterns between Nhe6 and markers of DRG neuron subpopulations, we found that nociceptors are the predominant expressors of Nhe6 within the DRG of wild-type mice. Behavioral assays with Nhe6 knockout mice demonstrated that impaired nociception develops primarily in aged knockout mice. This manifests itself as both a deficit in noxious heat sensitivity as well as an impairment of nocifensive reactions to high-intensity mechanical stimuli. Finally, functional investigation of Nhe6 knockout DRG neurons showed that nociceptors themselves are functionally impaired. Capsaicin, the pain-causing molecule from hot chili peppers, is an agonist of the TRPV1 channel. Calcium imaging experiments using primary dorsal root ganglia neuron cultures from Nhe6 knockout mice showed dramatic reductions in calcium response amplitudes following application of capsaicin when compared to wild-type mice. However, their response amplitudes following potassium chloride application were almost the same. Furthermore, immunocytochemistry on these cultured neurons demonstrated a marked decrease in TRPV1 expression at the membrane of nociceptors compared to wild-type mice. This functional reduction in TRPV1 expression, which was not due to a loss of nociceptors in knockout mice, was also confirmed in behavioral tests, wherein Nhe6 knockout mice showed a significant decrease in nocifensive responses following intraplantar injections of capsaicin when compared to wild-type mice. Hyposensitivity to pain, as is observed in CS patients, provides an urgent physiological issue due to its negative impact on the quality of life of those afflicted. Our work characterizes the neuroanatomical expression of Nhe6, sensory impairments in a mouse model of CS, and investigates functional differences between wild-type and Nhe6 knockout nociceptors, suggesting that part of the deficits are in fact due to decreased nociceptor excitability.